we'll end this first tour of the study of adaptation by looking at some studies where trade-offs, inescapable trade-offs between costs and benefits, play a central role in structuring the analysis. The first one of these cases involves a question about testis size variation in fruit bats. These are large bats that eat fruit for the most part, and they have a wide range of social ecologies where the groups in which individuals spend most of their time vary from just a few individuals up to hundreds, thousands, even tens of thousands in a few species. And it's also true of these bats that both absolute and relative male testis size appears to vary a lot. And this gave rise, long ago, to the hypothesis that the species that live in large social groups might have reason to invest more energy in making large, costly, heavy to carry around testes that would help males succeed in sperm competition, where the social biology of the species provides opportunities for females to mate with multiple different males, who are then inevitably engaged in a form of competition to outnumber the sperm of other males that mated with the female at the same time. We'll come back to this idea when we talk about the evolution of sex differences. So how can we can't do an experiment like the one that Eric Green did with the flies in our last example to test this hypothesis. What can we do? Well, we can use the evolved differences, perhaps, as a natural evolutionary experiment to test the hypothesis I just briefly sketched about the costs and benefits of having relatively large testes, which is to say of investing a lot of energy, food, and so on in developing large testes. So the pattern is, in fact, that relative log testis mass does go up as with social group size. So in this figure B on the left, the log, I believe it is to base 10 of social group sizes as given in this phylogeny and table above is on the x-axis, and relative log testis mass is given on the y-axis. That would be testis mass divided by body mass to just account for how big the members of the species are, and then the logarithm of that is taken to compress the range of them. And so this is obviously a strong positive relationship. Is it evidence for the adaptive hypothesis under consideration? Well, not in a clean way, and that's because related species are not independent points in a comparison of evolutionary outcomes. The species with large social groups tend to be closely related to each other. For example, in this clade near the top of the tree as it's drawn, and some of these other species with very small group sizes also are each other's close or relatively close relatives. So what are you to do? Fortunately, a few decades ago, phylogenetic methods were developed for comparing contrasts in group size, that is, phylogenetically controlled contrasts between lineages where group size seems to have gone up or down, and testis size seems to have gone up or down, with the contrasts calculated the same way in testis mass. This avoids the problem of overcounting the number of evolutions of small or large group size and small or large testis size. In effect, it the objects of statistical analysis become the branches inside the tree and what happened on them, rather than the current states of group size or testis size and body size in the species currently at the tips of the tree. And when those phylogenetically controlled analyses of evolutionary increases and decreases are done, the hypothesis is upheld. As you can see from this figure, it may be not quite as tight, but positive contrasts in group size tend to go with positive contrasts in testis size and vice versa. So we can conclude with some confidence that relative testis size does increase when group size increases and decreases when group size decreases by using, that is, the evolutionary history of this group of related species as the experiment, a natural experiment that is given to us to evaluate. And as we've said more than once, limitation by trade-offs and constraints always underlies every adaptive story. It's sometimes referred to as the no free lunch principle. Here's another example from Freeman and Heron. Heron and Freeman, your textbook. Larger flowers in this begonia are more attractive to bees. It's especially true for female flowers on the right. The flowers come in male and female. They look similar, but the bees try to avoid female flowers because they aren't rewarding to the bees, which are after nectar. So why don't female flowers just evolve larger size? Then the bees would have a harder time telling male from female flowers, and they would end up setting more seeds. Well, it turns out inflorescences like this with larger flowers tend to have fewer flowers because there is a trade-off between size and number, and that seems to explain why female flowers are no larger on average than male flowers. And the evidence and logic for this is laid out in Heron and Freeman in section 6 of chapter 10, which you can look at if you want to see the data and reasoning behind that conclusion. Here's another colorful and exciting trade-off involving guppies, species that many of us have known in our life. Male guppies, you may recall, are often quite brightly colored. Females like them to be brightly colored, that is, they tend to prefer brightly colored males as mates, and so do predators. So there's the basis of a very dramatic trade-off. And it's been known for some time that in Trinidad, where guppies are abundant, males tend to have fewer and duller spots in streams where there is a strong predator from the genus Crenocycla, which will be denoted capital C in the figures to come. Whereas in streams with a weak predator, rivulus, that's not terribly dangerous to the guppies, the males develop more and brighter spots. So to test the hypothesis that males really do face this trade-off between mating success and predator avoidance, John Endler did an experimental evolution study right here in our department. He set up artificial stream populations with guppies from a Crenocycla stream, that is from one of the streams with a strong predator in Trinidad. And there's where it took place, and the greenhouse is right above my lab, as it happens, above the third floor of South Biology. Then after 6 months, he added either Crenocycla, the strong predator, rivulus, the weak predator, or no predator at all. That treatment was called K for control, because C had already been used for Crenocycla, and he waited and let natural selection, again, this rapid adaptation that we have been talking about the last few days, actually tell whether the trade-offs worked as expected. And here's the amazing figure from his paper. Time in months, 20 months on the x-axis, average number of spots per male fish on the y-axis. Here at 0 months is where he established each of the stream populations, which were done in replicates. The average male spot number was 10. Then what happened over the next 4 months or 5 was an increase, quite a significant increase in spot number by nearly 2 per male, driven presumably by female preference for males with more spots, and no countervailing pressure for them to reduce their spot numberness and colorfulness because there were no predators. So it's at this point here where the curves diverge that predators are added or not. In the upper dark colored curve, it's rivulus, the weak predator. In the colored curve is a continuation of the no predator control. And then these are the streams where Crenocycla, the strong predator, was added. And of course, what happened was, as expected, that spot number declined dramatically and significantly in the Crenocycla treatments. It continued to climb a bit and then leveled off in the weak predator treatment with rivulus, and also in the no predator controls. So why did Endler include the K treatment, that is the no predator treatment, in the experiment? Here are 3 alternatives. I won't go through it now. I'll leave this again as an exercise for you to think through. Only one of these 3 statements is correct, and it makes sense under the circumstances. The other 2 really don't. So see if you can figure that out. It could reappear in a quiz or an exam near you in the near future. Here from the textbook again is an interesting observational study about adaptation under an unavoidable physiological constraint in a fuchsia from New Zealand. In this species, young, receptive, fertile, and rewarding to pollinators, flowers are green, and old, unreceptive, infertile, unrewarding flowers with seeds developing inside turn red. But then they stay on the plant for the better part of a week. The question that the investigators asked was, why not simply drop those old flowers? You don't need them to be pollinated, and they're not providing any reward to bees, and the bees know that they aren't rewarding. Why maintain them on the plant for 5 days? And no, it's not that red flowers attract pollinators. In fact, they repel pollinators who have learned long ago that they won't be rewarding. So here's a diagram showing the parts of these flowers with the ovary back here, where the seeds will be developing, and then after they open, they go through these phases of being green, which falls off after about a week. Then there's sort of an intermediate coloration, which is followed by bright red, which hangs on the plant for a full week or more sometimes. And then the abscised, that is the flowers that finally are dropped, they start dropping after, again, something like 4 or 5 days, typically, of the red flower being held on the plant. The answer, figured out by Kurt Lively and Linda Delph. Lively, you may remember, is the one who studied the sexuality of snails in New Zealand. We'll come back to that study again later. The answer is that some pollen tubes, after the pollen's deposited on the stigma, need the better part of a week to grow to reach the ovary, which is some distance away, some centimeters apparently away from the stigma. So dropping the flower during the pollen tube growth would reduce seed set. And the evidence the 2 found for this is that they just measured how many pollen tubes made it to the ovary, and took 3 days anyway from pollination before all the pollen tubes got there. Variation in the environment is probably very often a source of trade-offs. Lissy has given you a number of examples of this in the early part of the course, having to do with generalist and specialist adaptations. Here's another great study of this kind. It's again testing this general idea that being optimized for a particular environmental condition makes, like this hypothetical trait number 1, where some value of the environmental variable is on the x-axis and fitness or performance of the phenotype is shown on the y-axis. So trait 1 is relatively strongly specialized for this intermediate value of the environment, whereas trait number 2 is more flexible, more generalized. It does better at extremely low and high values of the trait, but not as well at intermediate values. So the general form of the question is whether a more flexible generalist phenotype will have a lower maximum fitness than a less flexible specialist phenotype. That's an implication of this general model of the trade-off between specialism and generalism. Here's a data set that supports the idea that such a trade-off is likely to occur. It shows real data on the maximum running speed versus the width of the performance curve, evaluated as like the standard deviation of the performance curve for speed, running speed as a function of temperature for a parasitoid wasp. The black points are individual wasps that were tested for their performance breadth. It's the log performance breadth in degrees centigrade versus their maximum velocity in meters per second. Again, the log of maximum velocity in meters per second. So both the black points and the colored points, which are the means of families, show significant variation in both of these measures, both the performance breadth and maximum running speed. And there is a negative relationship between them, meaning that the individuals and families that tended to show a better performance breadth, that is to say, a wider curve over which they performed reasonably well, tended to show a lower maximum running speed and vice versa. So that implies that, it's consistent with the hypothesis, that a cost of broad thermal tolerance is that your maximum performance suffers in exchange for that ability. All right, so that's a key idea in many studies of adaptation, and we'll see it coming up over and over again over the next few lectures. So the summary for this lecture is that adaptive evolution is ongoing in most species most of the time. There's no way to escape selection on most trait variation, and often, but not always, that selection ends up changing trait values. Stabilizing selection that tends to maintain an adaptation by reducing extreme phenotype values appears to be very common. Organisms have many features, however, that lack adaptive functions as far as we can tell if we look at the situation honestly and comprehensively and quantitatively. And the functions of some traits that presumably have functions can sometimes be hard to determine. Science is hard. Organisms, although breathtaking in their design and beauty, as we said, are, when you look closely, far from perfect. And the reasons include the no free lunch principle that we were just focusing on the last few minutes, trade-offs. That's for example, in the guppy color spots, etc., there's no optimal phenotype. It all depends on conditions. Pleiotropy, the you can't do just one thing principle, and the flower abscission problem in fuchsia is a nice illustration of that. Then there's just opportunity. The you can't get there from here, or it may be hard to get there from here principle, which arises because selection can only act on available genetic variation. If the variation doesn't exist to enable a phenotype to evolve, then the phenotype can't evolve. One obvious manifestation of that is that we are stuck with 4 limbs. It might actually be better if we had 6, like an insect. Then we could have 4 legs, like a centaur, and our 2 arms. And hey, we'd be good in all kinds of terrain and playing the piano and everything. But no, it's not to be, because we have no genetic variation for limb number, and we're very unlikely to ever get it. A very general limitation on adaptation is the genetic load, which we discussed earlier in the semester. Most genetic changes, anyway, are for the worse. Mutations happen. Locally maladapted alleles can migrate in from elsewhere, and on and on. This is one reason for stabilizing selection to always be an important thing. And finally, there's evolutionary lag. Environments are always changing, but populations are always fighting the last war, so to speak. They're better adapted to past environments than to present ones because evolution simply takes time. And you can see that pattern very clearly in the Darwin's Finch story, where thanks to the length of time that the Grants and their colleagues studied the population, we've seen them go now through repeated cycles of adapting to a situation that they were maladapted for, and then having to undo that adaptation when conditions return to a previous state. All right, that's enough about adaptation, and see you next time when we will talk about sexual selection and the evolution of sex differences.